Happy morning, children. I feel extremely glad to welcome you all for our revision program. And this lesson number 7, cell cycle, has got some 5 question answers which we'll discuss in our today's video. Carefully watch it, revise those answers, write in your notebook, and learn. After that you write a home test in your assignment notebook. Come on children, let's revise. Let's start with lesson 7, cell cycle. This lesson has also got 5 answers. We shall discuss them one by one. The 11th question, the first question would be, write any 3 significance of mitosis. Any 3 significance of mitosis. That is, we are going to mention the importance of mitosis. You have got some 8 points in your textbook. Out of those 8, we are going to write only 3. Genetic stability, growth, repair of tissues. So, genetic stability here, the daughter cells are genetically identical to the parent cell. So, that would be the first important significance. Second, growth. As multicellular organisms grow, the number of cells making up these tissues increases. So, this would be your second significance. Third one, repair of tissues. Damaged cell must be replaced by identical new cells by mitosis. This would make the third significance. So, 3 significance: genetic stability, growth, and repair of the tissue. So, these 3 points makes your first answer, that is significance of mitosis. Now, let's move to the next question. Differentiate between mitosis and meiosis. Question number 12. Differentiate between mitosis and meiosis. So, here the mitosis and meiosis is given in 2 different columns and you will be having 7 points. We will discuss one by one. Mitosis is made up of 1 division. Meiosis by 2 divisions. Okay. Here the number of chromosomes remains the same. In meiosis, number of chromosome is halved. That is, it is reduced. You will have only half of the number of chromosomes. So, it is halved. Homologous chromosome line up separately on the metaphase plate. They are present separately. Here, homologous chromosome line up in pairs at the metaphase plate. They line up on the metaphase plate, but here they are placed separately. Mitosis, homologous chromosomes do not pair up. See, similar chromosomes are not pairing up. Here, homologous chromosomes do pair and form the bivalent. They undergo pairing and form the bivalent. Chiasmata do not form. You don't have a cross section. Chiasmata and crossing over never occurs. Chiasmata and crossing over occurs. Daughter cells are genetically identical. They have a similar genetical information from the parent to the offspring, daughter cells. But your genetical cells, daughter cells, are genetically different. The last point, 2 daughter cells are formed, 4 daughter cells are formed. Very simple thing to remember, children. Only 1 division, here 2 division, chromosome number remains the same. It is halved, it is reduced. They are separately placed on the metaphasic plate. They are placed on the metaphytic plate. Homologous chromosomes do not pair. Homologous chromosomes, they pair, and chiasmata and crossing over does not occur. Chiasmata and crossing over occurs. Daughter cells are genetically identical. Daughter cells are not identical. 2 daughter cells are formed. 4 daughter cells are formed. So, this is about the difference between mitosis and the meiosis. Now, let's move to the 13th question. Give an account of Z0 phase. G0 phase. This Z0 phase comes under one of the stage in cell cycle. A quiescent stage called Z0. So, Z0 is called the quiescent stage, where the cells remain metabolically active without proliferation. Cells can exist for long period in Z0 phase. In Z0 cells, cease growth with reduced rate of RNA and protein synthesis. So 3 points are there for Z0. It is a quiescent stage. and the cell remains metabolically active. Cells exist for long period. The cell stops growing and the rate of protein synthesis is reduced. So this is about Z0 phase. You can also add one more point. It is one of the stage in the cell cycle. Okay, Z0 phase is not permanent. It is not permanent and the cells are very much dormant. So you got 5 points over here. So you can add one more point to it also. So 5 points are here. The Z0 phase is not permanent. The Z0 cells are not dormant. Okay. Now let's move to the next important question. Differentiate cytokinesis in plant cell and animal cell. Cytokinesis here refers to division of the cytoplasm. How does the cytoplasm divide in the plant cell and in the animal cell? That is what we are going to look into the cytokinesis. During cytokinesis, animal cell, a ring of actin filament forms at the metaphase plate. Okay, at the metaphase plate, you will have a ring of actin filaments. Okay, they contract and cleavage furrow is formed and the cell is divided. Whereas in plant cell, a new cell wall is formed. That is a basic difference. The cytoplasm divides by forming a ring. This ring is made up of actin filament. This actin filament contracts and cleavage takes place. Whereas in plant cell, a cell wall is formed between the two cells and the daughter cell is cleaved. So this is the basic difference between cytokinesis and the animal cell, division of plant cell and animal cell. We are now moving to the last question. Write about the packaging and diplotin and prophase 1. So prophase 1 comes under the meiosis first stage. In that the prophase 1 has 5 stages. Of the 5, the packaging stage and the diplotin stage we are going to explain. In packaging stage, the fully formed synaptic complex exits during packaging. The homologous chromosome thickens and become recombinant. So this is what happens in packaging. You can find in the diagram the chromosome, they become thickened and they come out. The process of chromosomal crossing over is crucial for accurate segregation, for holding the maternal and paternal chromosomes together. So this is about your packaging. Now we'll move to the substage. Diplotin. Diplotin is a synopsis. Ends with disappearance of synaptical complex. Homologous pairs remain attached at chiasmata and finally leads to diakinesis. We are not going to look into diakinesis, only this point. Diplotin synopsis ends with disappearance of synaptiminal complex and homologous pairs remains attached to the chiasmata. So this is what happens here and you got a crossing over. You can refer the diagram in your textbook. I hope you will all have revised this lesson. Learn again, write the answers in your notebook and make a home test of these answers and be prepared for your forthcoming exam. Thank you children.